Hello, my name is Hyongbum. I'm pleased to join this exciting conference. Today, I'll talk about predicting the efficiencies and outcomes of gene editing using deep learning. Let's review the way we evaluate gene editing tools. Uh, we first clone a guide RNA encoding plasmid, then we deliver guide RNA encoding plasmid together with the Cas9 encoding plasmid to cell by transient transfection. Then we PCR amplify the region using a common pair of primers. PCR amplify the target sequence region using common pair of primers, individual pair of primers. Then the amplifiers are subjected to deep sequencing or disabling assay to evaluate of in the frequency at the target sequence. But this conventional method can, using this approach, maybe experienced students can evaluate maybe 50 guided activities over 2 weeks, but not more than that. But we wanted to evaluate thousands of guided activities by one experiment. To do that, we changed the process a little bit. Instead of cloning guide RNA, one guide RNA encoding plasmid, we generated a guide RNA target library of 10,000 target sequences. To this library, we delivered Cas9 nuclease. Then to this Cas9 nuclease, then we amplified the target sequence using common pair primers. Then those amplifiers are subject to deep sequencing to evaluate tens of thousands of guided activities by one experiment. So to make the library, we start from the oligonucleotide. This is the oligo, which composed of guide sequence and target sequence. We initially used this oligonucleotide from another company, but we recently changed to changed to order oligonucleotide from Twist Bioscience, from which we can obtain much better quality of oligos. Anyway, we start from this oligonucleotide containing guide sequence and target sequence. And by Gibson assembly, we prepared a plasmid library. And from this plasmid library, we generated the lentiviral library, which was transferred to genetic T cell to make the cell library. Each cell in this library contain specific guided encoding sequence, and that cell also has corresponding target sequence. But nothing will happen to the target sequence until we introduce CPF1 or Cas9. In the presence of this CPF1 or Cas9, for example, CPF1, they will bind to the transcribed guide RNA, then cleave the target sequence depending on the guide RNA activities. Then if guide RNA activity is high frequency in there will be introduced in the target sequence. If guide RNA activity is low, then the target sequence will be intact. Then to amplify this target sequence, we use a common pair of primers. Then those target sequences are amplified deep sequencing. Then we analyze data. Using those high throughput method, we will evaluate thousands of guided activities by one experiment. but we wanted something more than that. We wanted to, we hope not to do any experiment. So we just wanted to predict activity by the target sequence composition. To achieve that goal, we use deep learning. So what is the biggest strength of deep learning is, as long as you have sufficient data, the performance of these deep learning-based computation models are often much better than those based on the conventional machine learning programs. Because we knew this, we and we next tried aimed to generate large amount of data of CPF connectivity. So for that, we generated about 15,000 CPF activities at 15,000 target sequences. And using this large data set, we generated this large data set and deep learning neural network composed of one convolution layer and 3 fully connective layers. Then we generate a deep learning-based model. This is the performance of deep learning-based model that we developed. X-axis to represent CPF activity predicted by deep CPF1. Y-axis to represent measured index frequency measured by individual experiment. As you can see, there's a beautiful correlation between two values, indicating that now we can predict CPF activity without doing any experiment. We can just sit in front of a computer and type in your target sequence, and the computer will do the experiment for you, and they will give the result within a minute. Using the similar method of high throughput evaluation and the computational modeling using deep learning, we tried to predict activity of base editors. In case of base editing, several kinds of outcome product can be generated. For example, in this target sequence, there are 3 target nucleotides. As within the editable window, each target nucleotide A can be edited to be G while remain unchanged. Using high throughput analysis of base editing outcomes and subsequent deep learning, we made computation models that predict proportions of these base editing outcome sequences. As you can see here, this is a measured value and this is a predicted value. Measured value and the predicted value, they look similar. But this is just for one target sequence. We tested our model using the thousands of target sequences. X-axis is a measured proportion, Y-axis is a predicted proportions of base editing outcomes. As you can see here, we observed a beautiful correlation between two values, indicating that our computational prediction models function very well. Not only predicting the outcome proportions, we also predict base editing efficiency. And we named the model AB efficiency or CB efficiency. And we test the model by, and then using test data generated from the library high throughput evaluation or endogenous evaluation, endogenous target and evaluation at endogenous target sequences. And at both tests, we observed a high correlation between the measured efficiency and predicted efficiency. And by combining these two models, we finally developed the deep ABE, which predict outcome, absolute outcome proportion, efficiencies, frequencies at the base edited cells. So this work was done by these two talented students, Myungjae and Higon. Now they are both postdoc, and Higon recently got a tenure track position, which is great news to me, and as well as to him. We also developed a method to predict primating efficiencies. So using the similar approach, we were able to predict primating efficiencies at integrate target sequences, shown in the left and shown in the right, and these target sequences. And let me switch gears. I'd like to talk about elapsed time recording using Cas9. The frequency of indels that is generated by Cas9 nuclease increases over time, as long as the expression levels of Cas9 and guide RNA are maintained. Different colors in this graph represent different pairs of target sequence and guide RNA. The rate of indel frequency increase are different depending on the target sequence compositions. Because the indel frequency increases over time, then can we estimate elapsed time from the observed indel frequency? To do this, we need to know the function that explains the relationship between indel frequency and time. This left graph shows indel frequency over time. We initially tried to find the function explained in the relationship between indel frequency and time, but which was difficult. Then instead of focusing on the indel frequency, we thought that maybe we can focus on the intake target frequency. The intake target frequency is 100% minus indel frequency. Then the intake target frequency looks like decrease exponentially. Exponential decay reminds me radiometric dating. Exponential decay has been used in radiometric dating, which have been used to estimate the elapsed time of rock or fossils. Like radiometric dating, we postulated that intake target frequency would exponentially decrease over time as long as stable levels of Cas9 and guide RNA level are maintained in the cell. So for stable maintenance of Cas9 and guide RNA levels, we first generate the Cas9 knock-in cell. Then we introduced a library of self-targeting guide RNA encoding sequence into the Cas9 knock-in cells by lentiviral vector. In self-targeting RNA, guide RNA encoding sequence is overlapped with the target sequence, which simplifies the system. Some people call it this self-targeting guide RNA as homing guide RNA. Anyway, the self-targeting guide RNA and homing guide RNA are the same. And using this system, we were able to maintain Cas9 concentration and guide concentration constantly within cell populations. Then we measured index frequency in this cell library over time. We confirmed that the intact target frequencies exponentially decrease, and the rate of this decrease was different depending on the target sequence compositions. So in some target sequences, the intact target frequency rapidly decrease. The half-life is only 3.3 days. But in some target sequences, intact target frequency decreases very slowly, making the half-life over almost 115 days. Because now we know the function that explains the relationship between elapsed time and index frequency is exponential decay, we next attempt to estimate elapsed time using index frequency. We have cultured 6 independent biological replicates of Cas9 knockin cells transduced with self-targeting guidance encoding lentivirus. We choose one replicate and calculate the half-lives of self-targeting guidance encoding sequence or target sequence. Then based on the calculated half-life, we then estimated elapsed time using the observed index frequency in another replicate. We observed that this estimation of elapsed time were highly reproducible across replicates. In this bottom graph, different colors indicate the replicate used the half-life calculation. For example, when we estimated time using index frequency observed in replicate A, using the half-life calculated from the data of replicate D, the mean relative absolute error was about 10%. We observed the levels of error was mostly lower than 10% across replicates, suggesting highly reproducible time recording. This is my lab members. I'd like thank all of my lab members, especially Myung-jae Song and Hee-gon Kim, who developed high-throughput evaluation method and computationally predicting models. And to those projects, also I thank Song-ro Yoon and Sun-woo Min from Seoul National University, who developed computation models. And the CRISPR-Clack study was done by Jie Park, talented PhD candidate in my lab. And I also thank all of my collaborators applied to Yonsei University and Hanyang University and other universities. Thank you for your attention. This is our institute. Then I will be happy to take any questions.